All right. So in this unit, Unit 7, what we're going over is meiosis. Our last unit, we primarily talked about mitosis and how it was asexual reproduction. So in this unit, we're going to switch gears and we're going to talk about another form of cell division where we're forming sex cells, which of course is going to deal with having two parents and making something that is different, having offspring that is different from them. So, of course, we have to know how sex cells are made and how it's possible to make a cell that's going to have half the number of chromosomes. Just real quick, I would like to go over why it's going to have a half number of chromosomes. It needs to have a half number of chromosomes because the typical human body cells have 46 chromosomes, and well, if dad gave all 46 chromosomes, then and mom gave all 46 chromosomes, then that would end up being 92 chromosomes. So that's the reason why we have to actually have a reduction there of the number of chromosomes to where the sperm cell only has 23 and the mother's egg cell only has 23, so that 23 and 23 would make 46, so that then the baby would have the right number, the fetus would have the right number of chromosomes, and for growth and development to occur. So that's what meiosis, what we're going to be talking about, is that reduction of the number of chromosomes. And then we're also going to get into what happens whenever there's some atypical alignment of chromosomes. We'll be getting that into this unit as well. So today we're going to talk about is meiosis. Now, there's really not as much as I'm going to get into as I did in mitosis, mainly because most of the stuff that happens in meiosis is the exact same. There's some differences there, and so like there's still a prophase, there's still a metaphase, there's still an anaphase, and there's still a telophase, but it actually has to do it twice in order to divide the number of chromosomes, in which we'll get into that. And so I'm going to show you some of those differences and also highlight some of the big differences you need to make sure that you know about. We're also going to be comparing and contrasting mitosis to meiosis, and that's going to be a key part of this unit as well. Is you're going to have to know how to be able to tell the difference between the two processes. All right. So, some facts about meiosis. Meiosis is for sexual reproduction, whereas mitosis was for asexual. Meiosis has two stages of division. So, whenever we talked about mitosis, we were talking about how it had prophase, metaphase, anaphase, and telophase. Well, that's only one stage of division. This will actually go through division twice. So, it's actually going to have prophase, metaphase, anaphase, telophase, and then it's going to do it again. And so they actually get that Roman numeral. So it'll say prophase one. The next stage of division would have prophase two in it. Metaphase one, metaphase two. They actually still look very similar. There's some small differences there that vocabulary wise are small differences, but as far as the actual scheme of things, it actually is a very important process that they are differentiated. The original parent cell is a diploid still. So just like in mitosis, we said that parent cell was a diploid. The same thing happens here. You're gonna have the difference is that now we're going to produce four daughter cells, and those four daughter cells are going to be haploid. If you notice, we don't put two in here; we just have a single N, which means that, of course, this and what that haploid means is that it's going to have half the number of chromosomes as that parent cell. And remember, that's a very important process because two halves are going to make the whole. And so that's the reason why we need that reduction. Also, a big difference here between meiosis and mitosis is the fact that we have four daughter cells. Four daughter cells, whereas mitosis, it only produced two daughter cells, and those daughter cells were diploids, whereas now we're having those haploids. And the daughter cells are genetically different from the parent cells. The daughter cells are going to be genetically different from the parent cells, whereas in mitosis, they were identical. So the daughter cells, like we said before, contains half the number of chromosomes, and that's why we call them a haploid. Is because of that. That will be a very important vocabulary term to know. Produces gametes. So before we produced somatic cells, and somatic cells were like your skin and bone. Now we're talking about producing a gamete, and a gamete is a general term for sex cells, in which the two specific sex cells are the egg cells and the sperm cells. So the sperm are made in the testes in males, and eggs are made in the ovaries in females. So why do we need it? So of course it's the fundamental basis for sexual reproduction. The two haploid gametes. So whenever we talk about the two haploid gametes, what we're talking about is that sperm cell and the egg cell. Whenever they are brought together, that's called, whenever they unite, that's called fertilization. And what they form is a zygote. And now we have an actual cell that has the complete number of chromosomes. Because whenever it was just the sperm and the egg, they only contained half the number of chromosomes. So whenever they come together and form that zygote, now they have the complete number of chromosomes, so that now that cell has all the instructions, and now it can actually start doing what it's supposed to be doing, which of course is going to form a child or anything that produces through sexual reproduction means. And so what I have right here in this image right here, this would be showing an egg cell. And then what we see coming into it are the sperm. And if you notice, only one sperm is allowed into it. And that's so that that chromosomal number will stay the correct amount. So this is talking about the two parts of meiosis. So if we notice here, we have meiosis 1. Meiosis 1 starts with that original parent cell, and then the chromosome number is there. It goes from the parent cell to the two daughter cells, and then those two daughter cells split again, and that's where we're actually getting the reduction. Now, if we notice right here, what we have is we have what's called homologous chromosomes. Oh, let me go back. We have homologous chromosomes. What those are is those are chromosomes that are the same size structure. That's actually what we have here at the beginning with this blue and this green. We have the ones that were the same size structure. So those would be those homologous chromosomes. So they have to separate. And of course, whenever that happens, now we have regular chromosomes that the chromatids are going to separate. And so meiosis 2 actually looks almost identical to mitosis, because you're dealing with the same number of chromosomes. You're actually not dealing with a homologous chromosome. We are going to get into homologous chromosomes, and anytime you see a description of homologous chromosomes lining up in the middle, well, because it says it's lining up in the middle, I know it's metaphase. And whenever it says homologous chromosomes, well, that tells me, that's a key term to tell me that we're talking about metaphase 1. All right. So talking about that reduction, so right here, right here where it talks about that prophase 1, that chromosome number is doubled. Well, that's what we see here from that first slide to the from that first cell to the second cell. Is if you look at it, it looks like two X's that are kind of stuck together. That's that homologous chromosome. And if you notice in metaphase 1, instead of it just being a single chromosome lined up in the middle, it's two of them that are back to back. So that's the homologous chromosome that we're talking about. Now, if you can see it, you might not be able to, but if you can, if we look, especially on this metaphase 1 one, we can really tell it. There's this green chromosome, but it has a little bit of blue on it. Just like this blue one has a little bit of green. That's going to be a major point that we're going to talk about here in just a second. That will be a major defining part of what's happening, and actually that happens during prophase 1. I just knew in this image we could see it better in metaphase 1, but this actually occurs during prophase 1. And what that's called is crossing over. So it's just called that because we're crossing over that chromosome information. They kind of trade places. So if you notice here, now this starts to look a lot more like mitosis. So we have the single chromosomes that are lined up in the middle, then the chromatids separate. But if you notice, we have two cells that are undergoing it. And we end way down here with four haploid daughter cells. So this might look really confusing, but one of the biggest things to know here is you're not going to have to know every single little thing about all of them. What you're going to have to be able to do is identify mitosis from meiosis. So the easiest way to do that, of course, is knowing that they're all going to start with a diploid parent cell, but mitosis only has two daughter cells at the end of it. And those two daughter cells are also diploid, so it would be 2N. Whereas in meiosis, it's going to go from one parent cell that's a diploid, and it's going to end with four haploid daughter cells that have half the number of chromosomes as the original parent cell. And then there's going to be a couple processes that happen along the way that we'll definitely have to know about, like crossing over, which I said on the slide before. So talk about these tetrads that form during prophase 1. The tetrads are those homologous chromosomes that form together. Oh, there we go. That's those homologous chromosomes that stick together. This is when, prophase 1 is when crossing over takes place, and it takes place on these tetrads, on these homologous chromosomes. This is a big point to note. So crossing over. So this is a great picture that I found. So right here we have two sets of chromosomes that have been duplicated. And basically what's going to happen here is they're going to actually come together. And whenever they do, you notice they get twisted up. But what happens right here? We actually start to see this crossing over happening. We see that crossing over happening there. And that's a very important process because of this right here. Crossing over happens during prophase 1. And the purpose of it is it creates genetic diversity in a species. Whenever a species doesn't have enough genetic diversity, that means that species is extremely easy to wipe out due to an illness or basically being able to, the whole idea of survival of the fittest. The survival of the fittest means that we need to have genetic diversity. We need to have crossing of that happening, and we need to have a big genetic pool there. And that actually, so that's what this is going to help make, is going to help create more genetic diversity within a species. This is a big part of what you're going to have to know, and it occurs during prophase 1. And then, of course, the results of meiosis, we need to make sure that we understand that. Meiosis produces gametes. The gamete is the egg and the sperm, the 4 haploid cells with one copy from each chromosome. The different combinations of those genes along the chromosome, that's what's going to be creating that diversity. And the reason why we have different combinations is because of crossing over. So now let's compare the two of them. So before we go on to that, please don't forget in meiosis, and it's going to have those 2 stages of division, so really you're going to have a lot of little facts to know about it. Don't forget, anytime you see the term homologous chromosomes, well, then that means you're talking about meiosis 1, that stage of division. All right? Otherwise, all the information we learned about during mitosis about how prophase, the nuclear membrane has to break down, the nucleolus disappears, spindle fibers form, all of that remains the same. So that's why it was so important for you to learn it the first time. If you don't remember some of those key things that happened, you need to make sure that you go back and take a look at that so that you would have some of that, still some of that information. So right here, I just put this in just a general chart like this, and just showing some of the comparisons between the two of them. Mitosis versus meiosis. The number of divisions, mitosis only has one division cycle, whereas meiosis has 2. Number of daughter cells, 2, and whereas meiosis has 4. Genetically identical. Mitosis, yes, they are. Meiosis, no. The chromosome number is the same as the parent. So right here, if you need to, make sure that you know that parent is a diploid. That means the the offspring in mitosis are also diploids. Whereas in meiosis, the parent, this is going to be half the parent, so the parent's still a diploid, but now the daughter cells, those four daughter cells, they are haploids. Where does it occur? In somatic cells. Remember, that's going to be your bone and skin. For gametes, that's going to be in your sex cells, sperm and egg cells. When does mitosis occur? It occurs throughout life. Meiosis is going to start occurring during sexual maturity, hitting puberty, things of that nature. And the purpose, the role of mitosis, the main role is all about growth and repair. Meiosis is all about sexual reproduction. Let's reduce the number of chromosomes. Let's also create some genetic diversity here so that whenever it adds with somebody else's who has created that genetic diversity as well, the species has a better chance of survival. And so that's the main points out of it. So right here's the review for it. So I asked you about some key parts about meiosis. And really looking at that slide that we just looked at, the previous slide, that would help you out with knowing some of those basic facts about meiosis. But I would still make sure you go back to that slide and look at some of those basic facts. So how many daughter cells does meiosis produce? 4 daughter cells. Why are the daughter cells different from the parent cells? Well, the daughter cells have half the number of chromosomes, but they're also different because they underwent crossing over. And that actually caused their chromosomes, their genes, to be different on each chromosome. What form of reproduction is meiosis? It's sexual reproduction. What are female and male sex cells? Well, we could either call them gametes, or we could be more specific, like, which I think this right here does imply that the females have eggs, whereas the males have sperm cells. How many chromosomes does each sperm cell have? It's going to have 23, or it should have 23 chromosomes. Same way with the female, it should also have 23. What is it called whenever the sperm cell unites with an egg cell? That's going to be fertilization. So that takes you through some of the facts. You need to also make sure that you remember some of that information about meiosis, as far as making sure that you pay attention to, of course, it has those 2 stages of division. During prophase, we have those homologous chromosomes, prophase 1, those homologous chromosomes that come together to form a tetrad. And that's when crossing over occurs, and the purpose and the reason and purpose around crossing over is to create genetic diversity. So next class, what we're going to be talking about is atypical chromosomal alignment and karyotypes. Okay?